So good afternoon, everyone, and welcome to Grand Rounds. This week is the annual DCAT lecture where we hand over Grand Rounds to Professor Ewan Pearson and the DCAT team. And as I said in my email, it's going to be slightly different this year, and Ewan will go into that in more detail, I suspect. This is one of the highlights of the year for me for various reasons. Mainly, first of all, the talks are always fantastic and I learn a great deal of stuff, but also it means I don't have to say very much because someone else chairs the meeting. So I get to shut up now and hand over. But what I will say is that this is Ewan's last year as being the DCAT lead. He's going to hand over to Anna Maria Choi, who unfortunately can't be with us today, to hand over the baton so not in person, but you know what I mean. So I'd just like to take the opportunity to thank Ewan for all his work with DCAT over the last, well, how many years has it been, Ewan?
I've lost count actually, Tom. I would think 10 years probably, yeah.
So all the work that he's done for DCAT over the last 10 years, I'm sure anyone who's been through the DCAT program, all the foundation doctors, the CROHN'S lecturers, the consultants who've benefited from his mentorship and steering through their research careers would like to join me in thanking him. Because it is a system which is envied across the country, near the world. Has been a fantastic system that really does churn out high quality research and high quality researchers. So Ewan, on behalf of me, Grand Rounds and everyone, thank you very much for your time and effort. And good luck to Anna Maria when she takes over. Big shoes. Ewan, I shall hand it over to you and I shall shut up for an hour.
Thank you very much, Tom. Hopefully that didn't work. Okay, let's start again. Good, okay. So thanks, Tom. And this very much is the highlight of the DCAT calendar. And I do think it's probably one of the highlights of the medical school calendar is the Dundee Clinical Academic Track Annual Lecture. Just a plug there for anyone who's interested, either clinicians in training or medical students who want to know more about academia, visit our website or email Anna Maria or myself. So as I said, it is the highlight of the year. This just to show the level of lecturer we have, and we've had now going back since 2012, and we've had over the years some brilliant talks. So as I said in the email that Tom circulated, we had asked and Raj had agreed to present this year's DCAT lecture, and Raj is an incredible speaker, a diabetes and endocrinologist, largely an endocrinologist, who I'm sure would have inspired us. For the reasons I said, in that the DCAT lecture is more than just a lecture, it's a visit, it's a meet the trainees. We've agreed that he will now present his lecture next year, which means we now have the alternative DCAT lecture, which I think will be probably better, to be honest, than these eminent lecturers we've had over the years. And what we've done is ask people at different stages of training to give a talk around the research that they've done. So Jamie will be talking to us around the time he spent as an academic foundation doctor, although he's now a clinical fellow. Iffy will be talking around work as a CROHN'S lecturer, although, as I'm sure he will tell us, he's just been appointed as a senior lecturer. And Esther will be talking to us around her more senior role as a Scottish senior clinical fellow. So at this point, I shall stop and I shall introduce Jamie. And Jamie is going to talk to us around his research with his title of Pharmacogenomics and Phenotypes, starting out in clinical research. So if I stop sharing, Jamie, do you want to take it on?
Yeah. Is that working?
Perfect.
Great. Well, thank you very much, Professor Pearson, for inviting me to speak today. My first involvement with the DCAT program was doing an intercalated degree here as a BMSc student. And that's paved the way for me to apply to the Academic Foundation program, which provides FY2 doctors with the unique opportunity to have 4 months carrying out research with no other clinical commitments. And it's the research I carried out during this block earlier this year that I'm going to be talking about. I have an interest in precision medicine and in cardiology, and Professor Lang kindly agreed to supervise a project for me in this area. After much discussion, Professor Lang had mentioned that several of his patients had had severe adverse effects from nicorandil therapy. This was not a new finding, but the BNF and the Tayside formulary both warn nicorandil use in patients with diverticular disease and those at risk of GI ulceration. However, despite this, patients were still coming to harm, so we chose to look into this issue in more detail. So what is nicorandil? Well, it's an anti-anginal agent that's currently prescribed second-line therapy, and that's on local prescribing and European Society Cardiology guidance. It acts to dilate coronary vessels, acting to offload the ventricles and improve coronary blood flow, and it has no impact on conductivity or myocyte contractility. However, it does have some quite severe adverse effects, and in particular, this is a mucosal ulceration, which can progress to both perforation or fistula formation. A previous study found in a cohort of patients with diverticular disease that those taking nicorandil had a 7-fold increased risk of developing a fistula compared to those who were not taking nicorandil therapy. In the Western world, approximately 50% of those aged over 60 years old have diverticular disease, and many of those have absolutely no symptoms. No formal screening is currently undertaken prior to the initiation of nicorandil therapy. So, pharmacogenomics. Examining the patient's genetic profile can sometimes help predict the risk of a patient experiencing off-target adverse drug effects. With cardiovascular therapies, this has been done before with statins and with ACE inhibitors, both finding genetic variants associated with myalgia with statins and ACE inhibitor angioedema. Fortunately for us, there was a recent GWAS study that was carried out which found a number of genetic variants associated with diverticular disease. Moving forward to our study, we thought that patients at an increased genetic risk of diverticular disease were more likely to develop adverse drug effects from nicorandil, requiring them to stop treatment early. So our aim for this project was to identify genetic risk scores associated with early nicorandil stoppage, which we thought would be a good surrogate marker for drug intolerance. We were fortunate to have access to GoDart's data, which is approximately 18,000 people, just over half of which have type 2 diabetes. And these patients preemptively consent to their prescribing biochemical and genetic data being used for research purposes. Looking at the diverticular gene associations, a recent biobank study had found 48 genetic variants associated with diverticular disease. And this study found that 27 of these variants were able to be replicated in a second European cohort. Looking at our GoDart's data, we had access to 23 loci and 42 loci respectively. And you can see here the SNPs that we used in our study. The ones with stars against them were the ones that were replicated in a second European cohort. So how did we go about creating our genetic risk scores? Well, we chose to make 2 genetic risk scores, one with all the replicable variants and the second with all the diverticular disease-associated SNPs. We then calculated patient-weighted genetic risk scores, and we used the natural log of the published odds ratio for each effect variant contributing to the score. We then added all of these values up to form the patient's weighted genetic risk score. For some patients, there were missing values, and we used the mean variant value for the cohort when this was the case. So I'm going to talk you through our selection process to determine our cases and controls. We can see that GoDarts gives us access to around 18,000 patients, and around 1,000 of those were prescribed nicorandil. We wanted to make sure that we were not including patients who stopped nicorandil for another cause other than intolerance. So patients who had their final prescription close to their death, for patients who had a recent myocardial infarction close to their last prescription, and for those who continued nicorandil therapy beyond the study date, were not classed as cases. That then gave 477 people, and we excluded those with less than 9 prescriptions, which equated to around a year of treatment, and made those our cases for our analysis. We also conducted an isosorbide mononitrate control analysis. Isosorbide mononitrate is another anti-anginal therapy that's not associated with ulceration. We did this in order to make sure that our findings were specific to nicorandil itself. We carried out univariate and multivariate logistic regression models to analyze this, and you can see the covariates that we included in our multivariate model. Our analysis were carried out for both of our genetic risk scores, and we carried out the same process in our isosorbide mononitrate cohort. So what did we find? Well, in that 23 variant replicable SNP score, we found a positive univariate association between our genetic risk score and early stoppage of nicorandil that reached statistical significance. When we carried this out with our multivariate model, the association strengthened, and it was even more significant than before. We then carried out the same analysis with the whole diverticular disease SNP score, the 42 loci, and we saw the same positive associations, but in both the univariate and the multivariate analysis, they were weaker associations that were not statistically significant. When we compared this to our isosorbide mononitrate control, we found weaker associations in all of our scores, and none of which were statistically significant. And the results for the univariate analysis are shown here, and our multivariate model below. So what do all these results mean? Well, we hope that the results show that patients with an increased genetic risk of diverticular disease are more likely to stop nicorandil treatment early, but not isosorbide mononitrate. And this goes against some of the initial case reports that predicted that adverse effects could not be predicted. There's a wide difference in the incidence of diverticular disease between Western countries and Asia, with, as I said before, around 50% of those aged over 60 being affected in Western countries, compared to just 0.5% in Asia. Diverticular disease recently has been shown to have a significant heritable component of up to 50% in Scandinavian twin studies. And interestingly, migrant studies show that when people move and adopt different diets, their risk of diverticular disease does not change. This is interesting when we realize that nicorandil first gained success in Japan in the mid-80s, and it was only when it was prescribed in France in the mid-90s that adverse events started to be predicted, started to become apparent. The main clinical implication of our study is that we hope that our genetic risk score can be used as part of a clinical decision support tool to help guide individual patient anti-anginal prescribing. And we've been very lucky to have our work recently published in this month's Clinical Pharmacology and Therapeutics, which is open access and everyone can hopefully have a read if they'd like. And I just wanted to finish by mentioning some of the great undergraduate opportunities that DCAT has given me. Throughout my project with my BMSc, I was able to have a first author paper publication, present this internationally, and it also set up further work through my elective. So I just wanted to let some of the undergraduate students listening know what great opportunities can come about from a DCAT project. Thank you very much for listening to my talk, and I'm happy to take any questions.
That's great. Thank you, Jamie. Do you want to stop screen sharing for a second, and we'll take questions? We're just a minute for questions before we move on to the next talk. And if you raise your hand, if you want to ask questions, and I'll start, Jamie. So I think it's a really neat study, and particularly clever to look at genetic risk score for diverticular disease rather than going down a sort of more pharmacogenomic route for the nicorandil itself. Your odds ratios were across the full spectrum of the GRS, so it's per unit of GRS. Did you look at the extremes of the GRS? Because obviously those with the highest extremes would be the ones that you would predict clinically might be useful to try and avoid nicorandil for. So
we didn't actually look at the extremes of it. We just looked at the trend over the score with that unit increase. But no, we had to assume quite a few different factors. It would be almost good if we could look at those extreme values and then try and correlate it with clinical cases. But unfortunately, we didn't quite have access to do that. That would be something that I think going forward, and if we were, hopefully as genomics comes more implicated in clinical practice, then we can hope to explore that in a bit more detail to see if we can find a cutoff that people, if they have a score above a certain level, would be at the greatest risk. Okay, I think something's just come into the chat. Oh, okay, that's fine. So I think what we should do is move on. We'll
if we have time at the end, we'll have general questions for everybody. So thank you, Jamie. And next up is Iffy, who's got a long title, All Things Great and Small, from Big Data to Small Trials to Understand Mechanisms of Cardiovascular Disease. So over to you, Iffy.
Great, thank you. Yeah, no, it's a pleasure to be here to present my work today, and thanks very much for the opportunity. So just a bit about my journey so far. First of all, so I graduated in 2007, and after my FY and CMT training, I was a cardiology clinical research fellow in Glasgow between 2012 and 2014, and got my MD based on cardiac MRI research in 2015. I had my cardiology number in 2014 and then transferred over following my enjoyable period of research to take an academic clinical lecturer job in Dundee in 2015. I was fortunate enough to be awarded a NES Chief Scientist Office clinical lectureship in 2017, which enabled me to go 50% academic and 50% clinical. And I had that post up until last month when I got a clinical senior lecturer post, having CCT'd in August of this year. So my research really is about, my interests are in doing clinical research to better understand mechanisms of cardiovascular disease in order to improve patient care, really with integration of knowledge from large data sets and following on clinical trials to impact clinical guidelines, hopefully. And I really just want to show a few examples of my research that I've been fortunate enough to be involved in over the past 5 years during my Screds post. So first off, this started off, as I say, my interest in research really was driven by my MD research. And this was a paper that we published in 2014 based on the cardiac MRI database in the west of Scotland that I was able to create. And this was looking at risk stratification of patients for primary prevention ICDs, so implantable defibrillators. So our current clinical practice is that we use defibrillators in patients who have obviously survived the previous VT or VF cardiac arrest, so secondary prevention, or we also use it in people who we think were judged to be at high risk of cardiac arrest, so people with heart failure and a reduced ejection fraction who've not had a previous arrest, so primary prevention setting. But in primary prevention, obviously we expose patients to device risks such as infection or inappropriate shock, and they may never use their device, and we also limit their life in certain ways. They have periods where they're not able to drive, for example. So I wanted to know whether we could provide evidence using our MRI data that might help risk stratify patients better. And what you can see here is by giving contrast in MRI, you can identify patterns of scarring in the heart. And in panel B, what we can see is a non-ischemic cardiomyopathy, where you have this sort of pathognomonic mid-wall scarring just here, which you see in non-ischemic cardiomyopathy quite often. And what we found in the study was that in patients who we adjudged to be at high risk, i.e. those who had this mid-wall scarring, or evidence of wall stress with a high NT-proBNP level, were at much higher risk of death or ICD activation. And in fact, in people who didn't have this mid-wall scarring, they had no events at 3 years. So taking this on in my Screds post, I was able to collaborate internationally and co-author meta-analysis where we pooled data of 3,000 patients with dilated non-ischemic cardiomyopathy, and we showed that mid-wall scarring was associated with a 4-fold risk of arrhythmia or sudden death, and actually a 5-fold increased risk as well in those with an ejection fraction above the normal cutoff that we would normally use for risk stratification. And particularly in the primary prevention setting, the presence of this mid-wall scarring was very much associated with risk. And this paper then was put into the guidelines in both Australia and New Zealand and in the US as well, suggesting that MRI could be used as a risk stratifier in this group of patients. So it's just an example of how we can influence practice with the sort of large data observational analyses, and hopefully the next European guidelines, this will be in there as well. So obviously this led to an interest on my part from risk stratification of patients for prediction or intervention sooner. Obviously, as a hospital doctor, the first presentation for a lot of patients that we see is in hospital with symptoms, and we can treat them in the presence of symptoms and signs before further deterioration. But obviously, this isn't particularly useful if the first presentation for the patient is sudden death. So I've shown how we can use imaging phenotypes to potentially risk stratify patients prior to events. And what I wanted to do was to use the rich seam of data available in Dundee to look at biomarkers and genomics, which could be used even prior to phenotypic expression to intervene on patients, and this could be the optimal time to do this. And this really was the focus of my NES-CSO lectureship. So you've heard a bit about SNPs from Jamie already, and this really is, the SNPs are a substitution of a single nucleotide at a specific position in the genome that's present in at least 1% of the population. And every person has about 4 to 5 million of these variants, and these can be associated with specific traits. So for example, you can see here, this person has a C in this position here, but at this nucleotide, they have a T, another person has a T, and these traits can each be associated with different things. So we explored genetic variants, these SNPs associated with coronary disease using Godart's data, and we used these genetic risk scores as Jamie has talked about. And we looked at this for the association between coronary artery disease and heart failure, and the two types of heart failure, reduced versus preserved ejection fraction. And this was published in Circulation last year. And the advantage of using this technique is that it's not, as opposed to traditional observational studies, it's much less liable to confounding, as obviously your genes are delivered to you at birth and you can't change them regardless of your lifestyle. So you can use this to as a level above traditional observational data, but still below traditional randomized controlled trials. And what we found was that, as you would expect from our observational analyses in black here, you can see that the presence of coronary artery disease was significantly associated with all types of heart failure. But what we actually found was that the genetic risk of heart failure, of coronary artery disease, was associated with an increased risk of heart failure with reduced ejection fraction, but not preserved ejection fraction. And this is important because coronary disease is very prevalent in the population, obviously, but it has potential implications for the risk of HFpEF and treatment as well. For example, actually revascularizing people with HFpEF and bystander coronary disease might not improve their outcomes in any way. I've also been fortunate enough during my SCREDS post to collaborate internationally as well. And this was one thing we looked at, which was looking at comorbidities in heart failure. Specifically in this study, we focused on age. And this was in part of the BioStat CHF collaboration, which is an EU-funded study, a European collaboration to identify opportunities for personalized medicine in heart failure. And so we looked in this study as to whether, which was published in the European Journal of Heart Failure, we looked at whether increasing forced up titration of heart failure medications was beneficial regardless of age. So normally in heart failure, we try and increase doses to the maximum tolerated based on clinical trial data. But as we know, clinical trials, patients don't often reflect the reality of clinical practice. And what we found was that increasing ACE inhibitors or ARB doses to the maximal target doses, so for example, 10 milligrams of ramipril, was beneficial in reducing mortality and heart failure hospitalizations, regardless of age. But in contrast, with beta blockers, we didn't see this. So although there was an improved mortality and heart failure hospitalization for younger patients, in older patients, those over 70, we didn't see that same dose-response relationship. And actually what we found was that middle doses were actually the best for older patients, perhaps because of hypotension or bradycardia that's obviously commoner as we get older. So this is an opportunity potentially for actually a sort of precision medicine approach in heart failure based on age. And obviously precision medicine is something that Dundee is very good at and have a strength in, and we've been fortunate enough to contribute to a consortium in heart failure, again in international collaboration with 65,000 heart failure cases, looking at the genetic basis of heart failure. And in this collaboration, we've been able to identify 12 genetic variants that have been significantly associated with heart failure, including ones associated with coronary artery disease and atrial fibrillation, but also ones associated with cell death and protein homeostasis that might identify future treatment targets for heart failure. And we're continuing to produce work on this, looking at heart failure subtypes and outcomes in patients as well. And we've had some follow-up work on this, and obviously, as you know, there's an interest in Dundee with diabetes and heart failure. And we did what's called a Mendelian randomization analysis, which essentially is fairly similar to the genetic risk score approach, where we looked at the association between diabetes, insulin resistance, and heart failure. And as you would expect, we found, similarly to the observational data, that genetic risk of diabetes and genetically determined insulin resistance were both significantly associated with the development of heart failure. But interestingly, we found this bidirectional relationship, where we found that the genetic risk of heart failure was also associated with an increased risk of development of diabetes, perhaps suggesting that actually heart failure itself can cause diabetes as opposed to just being associated with it. And this is just in revision in diabetes care at the moment, so hopefully we'll get this published next year. And taking this on further, I looked at the Tayside Echo database to look at this in our observational data. So what I did was to look at people without diabetes at the time of their echo, dividing the cohort into patients with heart failure, and then matching them with patients who had had an MI but did not have heart failure as a kind of active control, and then also control patients without heart failure or an MI. And what we found was that heart failure patients at the first diagnosis of diabetes had a significantly higher HbA1c at baseline, and despite similar levels of treatment, had worse control even at 12 months. We also had significantly fewer patients reaching target HbA1c, less than 50%, and reaching a target of less than 53 millimoles per liter. Again, suggesting that heart failure might be driving underlying insulin resistance, and the development of heart failure might be an opportunity to actually screen for diabetes, which is particularly important in light of the new diabetes drugs that we have. And again, following up with this diabetes and heart failure interest, obviously I've been lucky enough to be involved in randomized trials as well, which as I mentioned, are the highest level of evidence that we have. And this is just one example, a trial that we published recently, looking at the effects of SGLT2 inhibitors, one of the new diabetes drugs, in combination with diuretics in people with heart failure. And this was an important question because as these drugs have been shown to improve heart failure outcomes in people with and without diabetes, and I think they're going to be getting more and more commonly prescribed, and perhaps shouldn't even be thought of as purely diabetes drugs anymore, but perhaps could be heart failure and even CKD drugs. And what we found was that in combination with loop diuretics, empagliflozin actually caused a significant diuresis, but without causing a natriuresis here. And this is particularly going to be useful for us, for example, in the setting of patients with acute heart failure or diuretic resistance, where sometimes we want to add in an extra diuretic. And rather than adding in benzoyl-thiazide, for example, which can cause hyponatremia and significant renal dysfunction, it might actually be beneficial to use these drugs instead. So this is something that could potentially change practice in the near future, and something we're probably going to be seeing a lot more of in clinical practice. And then finally, I just want to talk about how genomics might actually impact on cardiovascular risk and patient care. Obviously, I've told you a lot about risk of disease, but how might this actually impact practice? So we know there's been a lot of evidence out there saying that clarithromycin, macrolide antibiotics, such as that, are associated with increased cardiovascular risk. However, the predictors of this are unclear and the mechanisms behind it are unclear. Nevertheless, there was a FDA warning just 2 years ago saying that clarithromycin was associated with this increased risk and it should be avoided in patients with heart disease. So we were interested in looking at using genetics and genomics to understand mechanisms, and we just had this paper published just this week actually. We hypothesized that the increased risk might only be present in certain patients, and we conducted this study looking at Tayside data of over 200,000 prescriptions in Tayside, of 170,000 of amoxicillin versus 35,000 of clarithromycin between 2004 and 2014 to compare risk of cardiovascular hospitalization. And what we found was that, as seen in other studies, there was about a 30% increase in risk of cardiovascular hospitalization, both short-term at less than 14 days and long-term up to a year, when patients were given clarithromycin rather than amoxicillin, even after adjustment for all other sort of typical risk factors. But interestingly, what we really found was that there was a strong marked increase in patients who were taking P-glycoprotein drugs, such as statins, digoxin, or calcium channel blockers, and amiodarone, with significant interaction compared to those without taking it, suggesting that this risk was actually driven potentially by P-glycoprotein. Obviously, I've mentioned the limitations of observational data, and we've looked at this further, looking at using a genomic approach. So we took two genetic variants, which are these up here, and we looked at people who had a lower genetically predicted P-glycoprotein activity. So it's essentially a proxy for being on a P-glycoprotein drug. And we found that those with the genotype, the AA genotype that was associated with low activity of P-glycoprotein, had a significantly higher risk of cardiovascular hospitalization when given clarithromycin rather than amoxicillin, whereas those with higher P-glycoprotein activity didn't have this. So that perhaps suggests the mechanism for for this interaction and for this increased risk with clarithromycin. So that's really just a few of the things that I've been involved in over the past 5 years, and then just a few general thoughts. So, I've really enjoyed it. It's really crystallized that this is what I would like to do for the rest of my career, and I've been fortunate enough, as I say, to get my clinical senior lecturer post. DCAT's been really helpful. There's excellent mentorship, and it's really key for someone starting out in their academic career. I've had excellent mentors, including obviously Professor Lau, my supervisor. The regular progress reviews, which have been really helpful in setting career goals and identifying areas of weakness and targets. And obviously the dedicated research time, which is flexible, is really useful as well as maintaining clinical training. The main difficulty is really just the fact that obviously it's only 20% research time, and your counterparts in England have 50% research time as well. So it's important to try and seek external funding for your research. And my approach really has just been to make the most of all available opportunities and see what works, to collaborate and publish. But it's important to focus on your clinical training, because you may wish that, to become a clinical consultant at the end of this. So thanks to all my collaborators and funders as well, especially Professor Lang, my immediate supervisor. Thank you as well to Professor Pearson, who's been real help for me over the time. I didn't actually realize you were giving this up, so thank you for all your support over the past 5 years. And really, I couldn't have done any of this without any collaborators, and obviously the most important chief investigators who who's spurred me on to do all this work, our family, obviously. So thank you very much.
Great, thank you very much, Iffy. And of course, many congratulations on your new appointment. Very exciting for us in Dundee to have you. Again, I'm conscious of time, but if anyone has any pressing questions, please raise your hand. And maybe I'll just ask, the PGP clarithromycin interaction story is beautiful, and I think highlighting how using genetics linked to biobanks is so helpful, because without the genetic data, you wouldn't have established the causality there. Do you see any of that mapping through to licensing or guidelines around use of clarithromycin, Iffy?
Yeah, potentially. I think it's a really interesting finding, and it's been picked up quite a bit already. In terms of a bit of sort of wider press interest and things. I think one of the things I've thought about is how we might explore it further to try and get some further insights into it, whether that's using probe drugs or studying biomarker levels, for example, after being given macrolides. So these are things that might further support our data. I don't think this maybe on its own is enough, but I think there's a lot more work that could potentially be done there, and that could really explore this and obviously provide insight for us.
Yeah. Okay.
A quick comment. I think maybe we should even look at other macrolides. The azithromycin is the one that also captures a lot of information, and I wonder whether that might be implicated. I think it's worth looking at that.
Yeah, definitely.
Great, brilliant. Thank you very much, Iffy. We should move on because last but certainly not least, we have Esther, who is also senior clinical lecturer with us and Scottish Senior Clinical Fellow, is going to talk us through LRRK2 kinase signaling in Parkinson's disease. So we're moving away from cardiology to neurology now. So Esther, over to you.
Okay, thank you so much for inviting me. And, we are moving away from cardiology and pharmacology, but not away from genetics, but rather from genetic risk scores and genomics and GWAS to really rare disease-causing mutations in Parkinson's disease. So this is I'm not based in a medical school, although I'm in the School of Medicine in Professor Lang's department, but actually I'm sitting about here. So my office is just the next window in the MRC protein phosphorylation unit. So here I have a slide about my journey. So I studied medicine in Germany. I graduated from the Ludwig Maximilian University in Munich. I did an equivalent to the academic foundation program in Heidelberg. This is where I met my husband. And one really influential person, Professor Anna Maria Choi, who really pushed me onto the course of an academic career. And then I moved to Scotland. I got into the Scottish Neurology Training Program. And I think it was actually when I gave a lecture in on a Thursday at 1 o'clock when Sarah Marshall came to me, who was the DCAT lead at the time, and she said, you need to apply for our Dundee Wellcome Trust Clinical PhD fellowships. And I had already basically been accepted into the Edinburgh program, but if Sarah Marshall has something on her mind, there's really like, there was just nothing you can do. And I think, like, you know, these kind of little things happen because that was when I then decided to stay in Dundee and started working with Professor Dario Alessi, who's the director of the MRC protein phosphorylation unit. I was then also, like, you know, awarded a Scratch lectureship. And, like, you know, during this time, obviously, like, also, like, you know, personal things are quite important. My first daughter was born Maya, and then Frida a few years later. And then I finished my clinical training in 2016. I think, like, another really important person has been Professor Jacob George, who supported me during this time as a NHS consultant with a fellowship that gave me time to continue working with Dario. And then luckily, almost 2 years ago, I got my my Scottish Senior Fellowship, which really has made a tremendous difference because I now have so much time to work in the lab and have my group here. And that is really important because the kind of work that I do, a lot of biochemistry, a lot of like mass spectrometry, is something that you really can't do in 1 day a week. So, but basically, like, you know, I work on Parkinson's disease. Parkinson's disease is really common. So 1 in 100 people over the age of 60 suffer from Parkinson's disease, 12,000 people here in Scotland, and 6 million people worldwide. So we don't really know what the cause of Parkinson's disease is. We know that it is advancing age, we know that it is environmental factors, and we know that it is genetics. And importantly, like, you know, probably opposed to other specialties, like, you know, we really have no test for diagnosis. It is the clinical diagnosis and the emergence of motor features that basically hallmarks the clinical diagnosis of Parkinson's disease. And by that time, quite a lot of neurodegeneration has already been going on in the brain. I mentioned that genetic factors play a role, and with regards to genetic factors, whereas, like, you know, you've heard mostly in the previous 2 talks here about, like, you know, the common SNPs and, like, you know, common variants. So, like, you know, my focus is on rare alleles that cause Mendelian, Parkinson's disease, and these are very, very rare, but they have a high penetrance. And basically, like, you know, it's these kind of genetics and these kinds of Mendelian genes that can give you a starting point to interrogate the underlying disease-causing mechanism on a molecular and cellular basis. And this is just, like, you know, to exemplify that, like, GWAS has also, like, given really quite a lot of scores, up to 90 so far, for Parkinson's disease. But basically, like, you know, my focus is, like, as I said, like, you know, on genetics, like, you know, rare genetic penetrant mutations, and as examples, GBA, alpha-synuclein, and in particular, the leucine-rich repeat kinase, which my whole life evolves around. But these genes give really, like, insight into the disease biology and mechanisms, and that can then lead to translational therapies. And the idea would be to really, like, stratify patients according to functional mechanisms that would then inform which clinical trial or research project is the most appropriate for a given individual. So my talk is on LRRK2 and Parkinson's disease. So LRRK2 is the most frequently mutated gene in Parkinson's. The G2019S mutation in the kinase domain is particularly prevalent worldwide. It causes about 1% of all sporadic cases. So if I take 100 patients with Parkinson's disease in Dundee, 1 to 2 of them will carry a G2019S mutation in the LRRK2 gene, and up to 4% of familial cases. But there is quite a lot of, like, you know, population specific differences. So for example, when you look at North Africa, 36 of all the familial cases of Parkinson's disease carry the G2019S mutation in LRRK2, and sporadic, so apparently sporadic cases, you know, like 42% of cases carry a LRRK2 mutation. And there has been quite a lot of progress to understanding what the LRRK2 kinase does. So basically, like, you know, LRRK2 is a really large protein with multiple domains, and then like here 2 catalytic domains, a ROCK core GTPase domain and the protein kinase domain. And what we know is that the clear-cut disease-causing mutations, whether they're in the kinase domain or in the ROCK core domain, they all have in common that they activate the LRRK2 kinase. And the consensus is that it is the increased activity of the kinase function of LRRK2 that causes disease. And a lot of work and progress really like has been spearheaded, you know, like by Dario Alessi here in Dundee. And he and in an international consortium identified the you know, first bona fide endogenous substrates of the LRRK2 kinase, which are RAB proteins, so a subgroup of RAB proteins, including RAB10. So RAB10 is what I will continue to work on in a little while. So basically, like the phosphorylation of these RAB proteins then really like impacts and guides, like, you know, then downstream effects. And that's like still like, you know, under investigation. But importantly, like, you know, there's an effect of the LRRK2 kinase mutations also on lysosomal dysfunction, which is really like an important scheme in idiopathic Parkinson's disease. And this is something that that we've also, like, you know, when you think of, like, you know, what can also, like, in addition to mutations, impact on the LRRK2 signaling pathway, so genes and non-genetic factors such as environment. Then, like, you know, we've identified RAB29, and more recently we've published that, like, mutations in BPS35, which is also like a Parkinson's disease causing gene, can activate the LRRK2 kinase. And then, so, like, you know, but it's really nice if you have all this evidence in mice and in heterologous overexpression system, but you really want to know, can you actually show these like single phosphorylation events in human biosamples? And then also, like, you know, is there, like, you know, what is the most appropriate biometrics? Is it blood? Is it urine? Is it CSF? Is it a brain sample? And what is the clinical utility thereof? So I think one of the contributions that I have done to the field is to really, like, you know, maybe like suggest that within blood, you know, like when you look at blood as a biometrics for Parkinson's disease into the LRRK2 kinase, it's probably, like, you know, neutrophils and monocytes that express high copy numbers of LRRK2 and one of its endogenous substrates, RAB10. Because obviously, if you want to interrogate the effect of LRRK2 on RAB phosphorylation, you need to choose a biomatrix that is homogeneous and prevalent and that you can easily look at. Because up to then, most people had just looked at PBMCs as a biomatrix, and you can see that quite a lot of cells that make up the heterogeneous population of PBMCs do not even express the LRRK2 kinase. So basically, like, you know, what we do is we isolate from whole blood either monocytes and neutrophils, and then basically, like, you know, lyse this and then like use this as a starting material for either mass spectrometry or quantitative immunoplotting. So this is like just like, you know, now one project that, you know, that I'm hoping, like, just like, you know, hopefully to submit by the end of this week. This was a study that was really, like, you know, about like 3 years in the making. And, you know, like, so we had a study population of 101 individuals, and these mutations, you know, like, they are really, really rare. So getting fresh blood from 101 individuals is really quite an undertaking. And I had to travel to Barcelona and to other places to basically, like, you know, obtain this. So we had 21 LRRK2 G2019S mutation carriers, 21 with the R1441G ROCK core mutation, 27 idiopathic Parkinson's disease patients, and 32 control. So we collected fresh peripheral blood, then we isolated peripheral blood neutrophils. We treated the neutrophils with and without ex vivo a LRRK2 kinase inhibitor, and then lysed the cells and shipped them to Dundee. And then I performed, on the one hand, quantitative multiplex Western blotting for phosphorylated RAB10, controlled by LRRK2. And then also, like, you know, we deployed a novel targeted mass spectrometry assay measuring phosphorylated RAB10, but not just like the relative ratio, but really the absolute occupancy. And the way we did this, we enriched for RAB GTPases, which are small proteins, by running a gel of all these, like, you know, hundreds of samples, cutting the band out, digesting the band, and then like spiking in stable isotope labeled heavy peptides as a standard of the phosphorylated and the non-phosphorylated RAB10 protein, and then basically measured this by mass spectrometry, and then did the data analysis. I will not show you all the results, but basically, or here like so, one of the people, one of the postdocs in the lab, so Ivona, traveled with me to Barcelona. This is like, you know, Eduardo Tolosa. He's one of the leading people, like, you know, one of these like tremendously famous people in the LRRK2 field. And then, like, you know, here we are in the lab, we occupy the whole section of the lab for 2 weeks. And then, like, you know, so here we had our cases together during this 1 week when we came. And this is just to somehow show you how the actual results look like. So this is the immunoplotting. This is just like, you know, of 10 individuals. And what you can see, this is the phosphorylated RAB10. And you can see by plain eyesight that some individuals basically like have high levels, whereas controls who do not carry one of the mutations in LRRK2 do not have such a significantly increase in phospho-RAB10 levels. And then importantly, if you treat these neutrophils ex vivo with a LRRK2 kinase inhibitor, the signal completely disappears, and that is proof that this is really like mediated by the LRRK2 kinase. And this is how this then looks like, and you can see like an over 3-fold difference between people who do not carry the R144.1G mutation, and then here who do carry the mutation. And then, like, you know, when you look at the same 10 individuals by mass spectrometry, you can see that, like, you know, clearly the R144.1G mutation carriers have a significantly increase in the peptides for phospho-RAB10. And basically, like, you know, we performed these 2 analyses on all 101 individuals, and this is basically just showing you the phospho-RAB10 for the occupancy, so the mass spectrometry analysis of phospho-RAB10, and what you can see is that R144.1G mutation carriers, either non-manifesting carriers, so who carry the mutation but do not as yet have Parkinson's disease, as well as people with this particular mutation and Parkinson's disease have a significantly increased level of phosphorylated RAB10, and the total RAB10 levels are, you know, are quite similar, and there's no statistical difference between them. And, you know, like also, like, you know, I've not shown you the all of the immunoplotting results, but when you here like look at the mass spectrometry and then the Western plotting by individual, you can see that there's actually quite a good correlation as demonstrated here. And to sum up, just give you a summary for this part of my talk, is basically like significant RAB10 hyperphosphorylation is present in LRRK2 R144.1G mutation carriers in blood neutrophils, irrespective of whether they have already like Parkinson's disease or whether they are still non-manifesting carriers, because the penetrance of this mutation is age dependent. And there's a good correlation between our multiplexed quantitative Western plotting assay and our novel mass spectrometry investigations. So I have a couple like now of other two other topics that I'm just gonna quickly mention. I'm also like very, very interested to somehow see whether the LRRK2 kinase pathway is important for any of the other monogenetic causes of Parkinson's disease. It has already been demonstrated for RAB29, but then here, like in a VPS35, that is like, as I said, like another gene mutation. And again, like, here I'm using monocytes, and here, just like, like mutation carriers, like of this particular mutation, 3 mutation carriers of the VPS35 D620N mutation were enough in comparison to either idiopathic Parkinson's disease or controls to really show that there's a significant increase, and that the VPS35 D620N mutation activates LRRK2 kinase activity by yet unknown mechanism. And that's basically in the lab at the moment what we are working to really try and understand what is going on. And I'm just going to touch on this one here. I'm also like working on inflammatory bowel disease, because LRRK2 is also, has been identified in a GWAS study in first Ashkenazi Jewish people, that LRRK2 is a risk variant in inflammatory bowel disease. This was then replicated in Ashkenazi, in non-Ashkenazi Jewish people. And then, like, the risk, the risk variant in particular for an increased risk for inflammatory bowel disease was, was in the kinase domain. So the N2081D variant is a coding variant in the kinase domain of LRRK2 that is associated with not only like Parkinson's disease, but also with Crohn's disease. And at the moment, I'm working with Inga Peter, who published these studies. And we have like so far 40 neutrophil samples and bowel biopsies of people with and without these risk or protective variants in LRRK2, and we are just about to analyze them. And this is like just an example of like, how we stratify this in a different setting other than human biosamples. So this is a really nice assay where we are using heterologous overexpression of like mutants and LRRK2 mutants in HEC293 cells, and that gives us an indication how active these mutants are. And I should probably also mention that I also enjoy very much working with the CRC and the neuroprogressive dementia network, and that I participate in clinical trials. So we're quite active in Dundee, and I'm quite pleased to somehow say that despite COVID, we've been able to carry on. So we participate in a genetic study where we basically look at how many people carry with idiopathic Parkinson's disease carry Parkinson's disease mutations. And so far, we've only started in December of last year, but we've already met half of our target. And we at the moment collaborate with Centogen, who is the commercial partner on this study, to really deploy our assay, this heterologous overexpression assay, where we can actually screen for the impact on kinase activity of all these mutants in LRRK2, so above and beyond the clear-cut disease-causing mutations, to see what the effect on LRRK2 kinase activity is. And this is again how these results look like. And this allows us then to somehow score which variants in LRRK2 have an increase in augmentation of LRRK2 kinase activity. And now, together with Centogen, we are looking to get biosamples, so human neutrophils and monocytes from people who carry these variants. And obviously, this would be evidence that these people probably would benefit from LRRK2 kinase inhibitor treatment in clinical trials. And my advice to others: career progression and success as a clinical academic comes in different shapes and colors. I think there's no one hat fits all. I think resilience, perseverance, and hard work. I've spent endless weekends, holidays in the lab working. Research environment and support is absolutely crucial. Investment into personal relationships and collaborations. And then I think something that's really, really hard, because knowing which opportunities to grasp and which to let pass, because it's really important to focus on something and bring it to an end, to a good end, rather than to basically spread yourself too far apart and basically then not be really an expert in one thing. And work-life balance, I think that's still something that I'm trying to figure out. So these are all my acknowledgements. And without Dario Alessi, without all the people who work with me, without the people in the medical school, Professor Lang, who's my mentor in the medical school, and everybody else, I think all my collaborators and patients, all this would not be possible. And so thank you very much.
That's great. Thank you, Esther. If you want to stop screen sharing. So we just have a few minutes left before 2. Any quick questions for Esther? And I'll start. And you touched upon this. I think the story was just starting to emerge at the end, but I was wondering around your phospho-Rab10 biomarker in idiopathic Parkinson's. So one of your earlier plots suggested it was a bit increased relative to controls. Is it only in those who carry those variants in LRRK2?
Yes, so I think we don't really see a clear-cut significant difference with Rab10. We've just published a paper, we've just submitted a paper where we established a novel assay where we are actually measuring 6 different phosphorylated Rab species in addition to other LRRK2 species to somehow get more of a differential signature and see whether there is something that can differentiate idiopathic Parkinson's disease from controls. But you know, like I have a lot of biomarker work in urine that I've not shown, where we actually now create a difference between controls, idiopathic Parkinson's disease, and these genetic types that fit into the LRRK2 pathway. So that's also quite exciting.
Great, thanks, Esther. I don't see a whole flurry of hands being raised, so I think given the time, we should probably...
Ewan, can I ask a quick question? Sorry, hands up.
Oh, excellent, Callum. Good.
Hi, Esther. Thanks. It's a very quick question. I was just wondering, have you checked whether the neutrophils have any functional difference in this mutation? Does the LRRK2 hyperactivity change neutrophil function?
No. So, you know, like we've not done any other assays, like, you know, apart from like looking at LRRK2 kinase activation in these. And, you know, like neutrophils, like, you know, are immune cells, and like, you know, the LRRK2 kinase is also involved in immune function, but we are not doing any other assays above and beyond, you know, looking at LRRK2 kinase activation. And
there's no immunological comorbidity that comes with Parkinson's?
There's a lot of interest in that, like, you know, so like, you know, already like inflammatory bowel disease, and like, you know, so I just got like, you know, biosamples from like, you know, hundreds of people from the United States, from the Michael J. Fox Foundation, and there was a particular emphasis to really like have very detailed clinical annotation of these samples, including like an extended immunological questionnaire for you know, for diseases, comorbidities, like, you know, medications and so on. So that's like, you know, that's a big interest, because also like, you know, is like, you know, are these factors also like important for idiopathic Parkinson's disease?
Okay, great, thank you. We should bring the session to a close. So can I just thank all three speakers? I think this was a really good session. I gave a wonderful insight for the academic, clinical academic track, from academic foundation through to people who've made it through the system and have got their tenured posts. So again, congratulations there to Iffy and Esther. And I'm sure, you know, this stands up as a high quality DCAT lecture, and I'm sure in years to come, all three of you will probably be at that level where you'll be invited in your own right to give maybe the 20th DCAT lecture, who knows. But thank you all, and thanks everyone for attending.
Thank you.
Thank you.
Cool.